Assalamualaikum warahmatullahi wabarakatuh and hello everyone. Let us continue on the last subtopic of recombinant DNA technology, which is the application of recombinant DNA technology. The application includes the production of transgenic plant, transgenic animal, production of insulin, and production of GMO, genetically modified organisms. But for this syllabus, we are going to focus on production of insulin only. For the learning outcomes, students should be able to briefly explain the application of recombinant DNA technology in mass production of insulin using cDNA. The general steps in production of insulin will be covered as well. Before we go further on the production of insulin, so let us discuss a little bit about the function of insulin. So insulin is one type of hormone that is produced by the pancreas. So insulin is responsible to control the blood glucose level. So when the blood glucose level is too high, so pancreas will release insulin. So insulin will send the signals to the liver, to the tissue cell such as muscle cell, fat cell, so they will take up glucose from the blood. So this will lower the blood glucose level back to normal. Okay, now we go to the next slide. So the problem arises when the insulin level in the blood is low. This is due to the body cannot produce the insulin. So not enough insulin to stimulate tissue to take up glucose. So this will cause high blood glucose level. And this condition may cause diabetes. Formerly, this disease was treated by the injection of insulin from animal source, such as pig and cattle. Unfortunately, the insulin from animal source are different in terms of amino acid sequence. And also, repeated injection of animal insulin may cause allergic reaction. So to overcome this problem, the insulin is produced using bacteria such as E. coli. And this technique is known as recombinant DNA technology. So this is an overview on how insulin is produced using bacteria or E. coli as host cell. As you can see in this diagram, the insulin gene is isolated from human pancreatic cell, while plasmid is isolated from the bacteria. Instead of we use the DNA as a donor, we use mRNA to obtain the insulin gene. Both of these insulin gene and plasmid are cut using same restriction enzyme, so that the insulin gene and plasmid are compatible to each other. Next, insulin gene is inserted into the plasmid to form recombinant DNA or recombinant plasmid. What happens next? This recombinant DNA or recombinant plasmid reintroduced into the host cell, which is bacteria or E. coli, by transformation. Then, this recombinant bacteria will undergo amplification to make many copies of them, as well as the recombinant DNA. The process occurs in the fermentation tank. Before the extraction and purification of this human insulin, recombinant bacteria will undergo screening to identify the recombinant DNA with the human insulin gene. As the conclusion of the overview, so production of insulin has 5 steps. So in here, production of insulin is using the cDNA. We will discuss later about the cDNA. So the first step is isolation. Second step is cleave or cut. Third step is insertion. Fourth step is transformation and amplification. And last step is screening. As I mentioned earlier, the insulin gene is obtained from mRNA instead of DNA. This is because of the difficulty in finding one gene among all the genes in the nucleus. Next, because of mRNA does not contain intron. Intron is a region on DNA or RNA that is not code for any amino acid. So means mRNA contain exon only. So because of this, mRNA consists code for a specific target gene. So what is the benefits of insulin produced by genetic engineering or recombinant DNA technology? So first benefit is insulin produced is safe since the source of insulin comes from human. So since that, the insulin also compatible and produce non-allergic to human. Third, it follows the Sharia compliance production. And number 4, of course it is cheaper. And number 5, the insulin can be produced in bulk or large quantities. Now we go to the steps in production of insulin using cDNA. First, mRNA code for human insulin is isolated from human pancreatic cell. Next, reverse transcriptase, which is enzyme, catalyze the formation of first cDNA. cDNA means complementary DNA, which complement to this mRNA. Third, mRNA is degraded by enzyme ribonuclease. So this is the degraded insulin mRNA and it leaves the single cDNA strand. Next, single-strand cDNA is synthesized by DNA polymerase forming double-stranded DNA. Next, what happened to this cDNA? So this cDNA will undergo adapter ligation by adding restriction sequence at both ends of DNA. So as you can see, these are restriction sequence. Okay, next, plasmid also is isolated from E. coli. And this is the cDNA just now. Both cDNA and plasmid are cut using same restriction enzyme. So this is the open plasmid, okay, and this is the cut cDNA. And both produce complementary sticky end. Next, what happened to this cDNA? This cDNA will be inserted into the plasmid. And then DNA ligase catalyze the joining of cDNA and plasmid, forming recombinant plasmid or recombinant DNA. Okay then, the recombinant plasmid is transferred into the host cell, which is E. coli, by transformation. These recombinant bacteria or transformed bacteria are amplified. Next, the recombinant bacteria undergo screening to identify the recombinant plasmid using blue-white screening. In here, the white colonies have recombinant plasmid. So these colonies are cultured. The amplification of recombinant bacteria occurs in the fermentation tank. In here, the insulin gene will be expressed to produce insulin. So these are the human insulin. Lastly, the human insulin is extracted and purified from the E. coli. So this is the product. So I hope all of you enjoy learning this topic. You may answer all these 3 questions. Thank you and see you later.